or Anna. So, okay. Can I start, or you will have a countdown? First of all, I would like to thank FESPE for allowing us, the Brazilian Pouring Club, to organize this symposium on signaling in the aging and disease brain. We'll have three presentations. The first one, age-dependent morphofunctional remodeling of astrocytes by Professor Alexei Semyanov from the Russian Academy of Sciences, Moscow, Russia. Then we will have a second presentation by Professor Yong Tang from the Chengdu University of Traditional Medicine in China, entitled Pruinergic Signaling in the Depressed Brain. And then finally will be my presentation, pruinergic control of neurogenesis and neurodegeneration in Parkinson's disease. I would like just to quickly to present the speakers, invited speakers. The first speaker is Professor Alexei Semyanov. He has a PhD and a habilitation from the University of Puchino in Russia, when he went on as a research fellow and senior research fellow at the University College of London, and he is actually the deputy director of the Russian Academy of Sciences and correspondent member of the Russian Academy of Sciences. He made very important contributions to the understanding of extrasynaptic signaling, neuroglial communication, and astrocyte signaling in the aging brain. Then the second speaker is Professor Yong Tang. He has a master and a degree and a PhD in acupuncture from the Chengdu University of Traditional Medicine. And then he had a postdoc at Sichuan University, also in China. And he was an honorary member of the Jack Stat Method and Research Award at NIH. And nowadays, Professor Yong is professor at the Chengdu University of Traditional Medicine. He's the vice dean of the School of Acupuncture and Tuinia, and he's the co-chair of the China Pouring Club, associate editor of pruinergic signaling, and he involves very much collaboration in the International Collaborative Center on Big Science Plan for Pruinergic Signaling. So, thank you very much. I would like to announce the first speaker, Professor Alexei Semyanov. Please, could you start your presentation?
Okay, so I will share my screen now, or it's shared already. So can you please tell me if you see my presentation? Excuse me, can you see my presentation?
Yes, yes, I see it.
Okay. Thank you very much for invitation, Henning, and introduction. It's my greatest pleasure to speak at this meeting. This is the first time for me to participate in Brazilian meeting, and I hope we will continue our collaboration, and maybe when pandemic finish, we will be able to meet in person. And today I will talk a little bit about our research in the lab. So a little bit, that means one of the projects which we carry on, and this is age-dependent morphofunctional remodeling of astrocytes. And I will start with a couple introductory slides, and I would like to show you this image, which probably most of you have seen in textbooks. This is very famous image showing the organization of the brain. So this is the part of the neuron, dendrites, synapses, synaptic connection, and this is illustrates synaptic organization of the brain, connections between neurons. And there is something wrong in this image. This space between neurons is illustrated as empty. And in fact, we know that neurons are not only cells in the brain, there are many different types of cells present there. And when we talk about physiology of an organ in our body, whether it's lungs, liver, kidneys, or something, we always explain its function from the point of interaction between different cell types. When people think of the brain, at least that was the case for most of last century, people thought of the brain as a large neuronal network and tried to explain all the function of the brain as would be neuronal interactions only. Few decades ago, people start to think of other types of interaction, of neuroglia interaction, of glia-glia interaction with the blood vessels and everything, and it turned to be very important. And now everyone knows that that's the case. And there was several different types of communication suggested. For example, many of you know about multipartite synapse. It started as a tripartite synapse, but the new parts added. So this is presynaptic neuron, postsynaptic neuron. There is some adhesion molecules. There is astrocyte, microglial process, and this system works together. So this modulation of synaptic transmission through different other elements of the neurous tissue. Also, neurogliovascular units. So when there is interaction between vasculature, endothelium, myocytes, and pericytes, glial cells, and feet of astrocytes, for example, and neurons. So this together, this system regulates local blood flow. Also, interaction happen between neurons outside of synapses, so-called extrasynaptic signaling, when a neurotransmitter leaves synaptic cleft and targets extrasynaptic receptors released by astrocytes or something. Volume transmission, when the larger territories of the brain actually targeted by diffusion of neuromodulators or something like that. And these are all different elements of the same system. And very recently, we published a paper with Alexey Verkratsky from Manchester University in Trends on Neuroscience, where we suggested to unify all those concepts with the concept of active brain milieu. So whether all these elements are just bricks which are integrated together and interacting with each other. And what you can see here, these are dendrites, belong to neurons. This is astrocytic branch with leaflets. This is astrocytic processes, capillary, extracellular space, microglial processes. And this is the milieu where they're all different elements interacting in a very complex way, and integration of this activity brings us the function. The function of the brain is not only neurons, but all different cell types. And of course, it's very interesting and important to see what happens to different elements of this active milieu during the lifetime of the animal or human being. Of course, we know something about neurons, but here we wanted to look what happens to astrocyte. This is one of the components of this active milieu. And we looked at the astrocytic morphology. So we used brain slices of mice, and then we patched astrocyte, loaded astrocyte with fluorescent dye, obtained these tags. Then we performed reconstruction, 3D mask, and then traced branches of astrocyte, and then performed show analysis. So these are spheres. This is 3D reconstruction, so it's a 3D show analysis, and we've seen how many intersections these concentric spheres made with the astrocytic processes. Then we looked at the astrocytes at different ages in animals. We took 3 groups of animals: young animals, 2-3 months old mice, adult animals, 8-10 months old, and old animals, 20-24 months old. and these are intersections. You can see that young animals have less intersections than adult animals. So when we grow, astrocytes also grow, and we'll experience astrocyte growth. But when we get older, well, not we, but mice, we're talking about mice experiments. Mice get older, the astrocytes shrink, they decrease, and the number of intersections decrease. Here is the summary of what I just said. Number of primary branches, with branches which emanating from the soma, didn't change significantly, but there was a trend also. But the maximal number of intersection, maximal number of branches increased from young to adult and decreased in old animals. So there is this biphasic change. Same happens to the branch length. So the first branches grow and then they start to decrease again. Then we looked at the territorial domain occupied by astrocyte as a projection of astrocyte along the axis, and this is like a shadow of astrocyte. And you can see young, adult, old, and again it increases this territorial domain of astrocyte and then start to decrease with old animals. There is no significant difference between young and adult, but there is a clear trend, but very clear drop in the territory of astrocyte in old animals. Then we looked at the territory and big branches, but astrocyte has a lot of very small processes which called leaflets, and we cannot resolve them with optical imaging because they're beyond the optical resolution. So we have to use other methods or indirect technique. And here we applied indirect technique, which is called estimation of volume fraction. Volume fraction occupied by fine processes was determined from their fluorescence divided by fluorescence of soma. Astrocytic soma occupies 100% of space, so this is 100% volume fraction. And when we divide the fluorescence of unresolved processes, we can see what fraction of volume they occupy too. So this is the astrocyte in adult and old animal, and there are several cross sections made through soma, and you can see them. So this is fluorescence of soma taken as 100%. Sometimes we cross branches, which are like a small peak, and the rest is actually a volume fraction of fine and resolved processes. So then we extracted removed soma from analysis, we removed branches from analysis, and then we averaged all this fluorescence for each cross section, and we obtained this volume fraction. And again, you can see that in adult animals, volume fraction of astrocytic processes about like 5% of the volume. In all animals, it's like 2.5, so it's twice decreased. So astrocytes become very loose, so it's dystrophic astrocyte with the age. And such changes in astrocyte might be associated with the change in number of astrocytes. We stained astrocyte with the sensor, not the sensor, the astrocyte-specific dye, sulforodamine 101, and look at the number of astrocytes. And we didn't find any significant difference in between adult and old animals. As you can see it here. And then we looked at the coupling of astrocytes to their neighbors. So neurons connected to each other with synapses. Astrocytes connected to each other through gap junctions. And gap junctions allow from diffusion of small molecules from one cell to another cell. And when we patch clamp the astrocyte, we load astrocyte with fluorescent dye, and this dye start to diffuse and get into the astrocytes which are connected to this particular astrocyte through gap junctions. And then we can just count the number of astrocytes coupled to the astrocyte of the interest. And then we discovered that in adult animals, the coupling of the astrocyte with the neighbors is much higher than in old animals. So astrocytes in old animals get smaller, probably lose connections to each other. So astrocytic network, astrocytic syncytium is also disrupted. So the communication between astrocytes disrupted. And what could that mean for functions of astrocytes? We started to look at their electrophysiology, the electrical properties of astrocytes. Astrocytes electrically non-excitable cells, but of course they have channels, they have transporters, and this all can be measured with electrophysiological means. So what we started with, we look at the current voltage relationship. So what we've done, we voltage clamp astrocytes, and then we gave the steps of voltage and measured current in these cells. And then we plotted this current voltage relationship. And we found that it became shallower, this relationship in astrocytes from old animals. And this indicates that input resistance in this astrocyte increases. And here's the summary of this data, adult and old. You can see a significant increase in input resistance in old animals. So which might reflect the fact that the astrocyte gets smaller. When astrocyte gets smaller, they have less membrane. And when they have less membrane, they have less channels which can be present in this membrane, and input resistance increases. So conductance decreases, input resistance increases. So this is the first quite interesting observation. Then we start to look at the other parameters which we can assess with electrophysiological methods. So we recorded transporter currents. Glutamate uptake in astrocytes is electrogenic, and when we stimulate synapses, so again we're working with slice, we stimulate glutamatergic fibers, and then we record current in astrocyte, and this current is mediated by uptake of glutamate because it's electrogenic, and we can measure this current and estimate how much glutamate is taken by the astrocyte. And we performed an experiment in 3 types of stimulation: single stimulus, 5 stimuli, and 4 stimuli with 50 hertz. And then we subtracted current to 4 stimuli from current to 5 stimuli. And so we get only one remaining transporter current, which corresponded to 5th stimulus. So, and then we compared the response to single stimulus to response to the 5th stimulus in the burst stimulation. So, and this way we can estimate activity-dependent changes in transporter current. And what we observed was quite interesting. Firstly, we found that there is activity-dependent facilitation of transporter current. And that's what we can see. This is transporter current to a single stimulus, and this is transporter current to 5th stimulus. And the transporter current to 5th stimulus is larger, both in adults and old animals. And there is no significant difference. This suggests that there is synaptic activity-dependent facilitation in both cases. Probably of glutamate release. But then we looked at the decay time of transporter current, and we found that decay time was longer in old animals than in young animals. So activity-dependent change was more clear and more expressed. And this suggests basically that neurotransmitter start to escape from the synaptic cleft in old animals and less efficiently removed by astrocyte. And this is obvious because when astrocyte shrinks, it becomes dystrophic, it removes processes from synapses, and basically glutamate start to diffuse. And this process called spillover, and this is negatively known to affect negatively synaptic plasticity. And synaptic plasticity we can estimate. So we measured here long-term potentiation. We recorded field EPSP, field excited postsynaptic potentials, in hippocampal slices in CA1 stratum radiatum. We stimulated glutamatergic fibers, Schaeffer collaterals, and recorded field potentials. And then we gave a high-frequency stimulation to elicit LTP, long-term potentiation. And the magnitude of this long-term potentiation was larger in adult animals than in old animals. So basically, in old animals, plasticity reduced. And this is the summary of this data from this endpoint, 60 minutes after the stimulation. Okay, how can we prove that this is related to glutamate uptake, which is reduced in old mice? So we blocked glutamate transporters. We block glutamate transporters with a drug called TBOA, and this and upon blockade of transporters, the difference between long-term potentiation disappeared. Long-term potentiation decreased in both cases because glutamate started to leak, but the effect was more profound in adults than in old animals, because in old animals already efficiency of glutamate uptake reduced. Okay, so we've done this experiment in mice, and this is a paper which recently came out in AGEING Cell. So you can look at this paper if you're interested. But also we continue working on this, and the question was how this phenomena occur in human. And of course, we cannot do all experiments in human, which we can do in animals. But in principle, we could access to human tissue if we collaborate with clinicians. There is a situation when humans develop glioblastoma. This is very unfortunate situation which requires surgical removal of this glioblastoma. And here is one of the example showing that glioblastoma is present in sort of subcortical area. So this kind of cortex, and this is in the brain. There is a region where glioblastoma has to be removed, and then neurosurgeons get to this glioblastoma, and they remove a little bit of a tissue on the way from the cortex, which is not affected by glioblastoma, to get access to these cells which they remove. So they remove it, and they give us a little bit of this so-called access tissue, and then we can perform experiments. So we can put under the microscope this tissue while it's still alive, and also load it with the pipette fluorescent dye. And that's what we have done here. These are several examples of astrocytes already reconstructed the same way as I explained to you before. For 3 people aged 33 years old, 45 years old, and 60 years old. And you can see here, again, there is some kind of trend that at first astrocytes are large. At 34 and 45 years old people, astrocytes large, but when we take astrocyte from 60 years old person, the astrocyte gets smaller, and this is the Scholl analysis, 3D Scholl analysis, number of intersections and distance, very obvious, very clear effect. And then, of course, we didn't get all the standardized ages, because people get glioma in different age, and depending on when the surgery happens and so forth, we get tissue from people of different age. And then we summarize this. Each point corresponds to one patient in the hospital who received this neurosurgery. And then you can see this result of Scholl analysis, the number of intersections at the age of from 30 to approximately 45. There's no much difference. So there is no much difference. And then after age of 45, approximately, there is a decline in the number of intersection, number of processes. And this is the summary of the data. And we also can look at the length of the processes. There is also more or less a clear, steady period after 45 years, and then the decline starts as well. And then we looked at the main area. This is again 34, 60, same patients. And we see here is a more or less steep for all the periods. So there is no kind of steady state from 34 to 45 or something. There's a decline, more or less, with the age. And then we looked at the astrocyte coupling. In human, we've seen a lot more coupled cells than in mice. So the astrocytes much better connected in human brain. And then what we can see here, there is no obvious changes with age. So they're all very variable, this number of connected cells in astrocytes in patients of different age, but there is no significant decline. Also, there is a trend, but there is no absolute statistical confirmation of this trend. And at the end, we performed a very interesting technique, which is called Raman microscopy. Raman microscopy based on the scattering of the light at the molecules within tissue. We can use a fixed tissue, we can use a live tissue, and then we can basically stain astrocytes with fluorescent antibody, or even make them to express some fluorescent protein, and then we can do Raman microscopy or from this particular cells, and then we get spectra like that. Sorry, there is a description here which is gone somehow. This is cytochromes, reduced cytochromes, mitochondrial cytochromes. This is phenylalanine, lipids, proteins. And then what we can do, we can look what happens to relative concentration of these molecules with the age. And what we've done, we look at the ratio of proteins to lipids with age, and we observed a decline with the aging. This is in astrocytes specifically. So we can see that there is metabolic change. So either more lipids or less proteins, but there is remodeling of the cell in terms of metabolism. And here is the number of cytochromes, reduced cytochromes in astrocytes, which there is a trend, but it didn't reach significance, very variable measurement. But if we get more patients, we get more tissue, for example, then probably we can get statistical difference. Also, working with human tissue, extremely difficult. And on this, I would like to make conclusion, because I try to be on time. And what we observed that with aging, astrocytes become atrophic. They start to lose processes. They get smaller, and they probably retract from synapses. And this is the adult situation when there is a synapse, which I mentioned, like a multipartite synapse. So this is presynapse, postsynapse, and there is astrocyte. It's very efficiently removes glutamate, potassium released in the synapse, and activates, this glutamate activates postsynaptic AMPA and NMDA receptors, triggers good synaptic plasticity. But with old age, astrocytes lose connectivity through gap junctions, and they shrink. So they open the space for neurotransmitter to escape from synaptic cleft. And in this case, neurotransmitter can reach extrasynaptic receptors, for example, NR2B receptors or NMDA receptors, and this in turn would reduce magnitude of LTP. So that's our sort of view on the situation. And this is the description of what's going on. And next step would be, of course, to find the treatment, how to deal with this, how to make astrocyte grow again. And there are different approaches, like active lifestyle, caloric restriction. It's a special diet when we reduce calories. It's all trigger astrocytic growth. And maybe at some point we can find a way how to intervene with this through sort of targeting molecular pathways in astrocytes. So basically come with the medicine. But this is the first observation on this way. And on this, I would like to conclude my talk, and I would like to thank people whom I'm working with. This is a good image, but photo of the lab, but unfortunately there is not everyone present here. It was just an institute. We're taking this occasionally. And these are the lab members so far who contributed to this work. Alexander Popov, one of the main contributors, Alexey Brazha, Nadezhda Brazha, Oksana Sutyagina, and our collaborators. These are neurosurgeons, Igor Medyanik, Alexander Yashin, whom we're collaborating with. And on this, I would like to thank for listening, and I would stop my presentation and would be happy to take questions.
Thank you very much, Professor Semyonov, for this excellent presentation. So questions will be at the end, and I would like now to ask Professor Yong Tang to continue. Thank you very much.
Okay. Good morning, everyone. Dear Henny, can you hear me?
I do.
Okay, here we go. Thank you so much for a kind introduction, dear Henny. And I'm also most grateful to you to organize this wonderful meeting. It's my great honor to be here to give the lecture at the annual meeting of FESBE 2021. My topic will focus on cholinergic signaling in depressed brain. As we know, ATP is often called the energy currency of the body because, in a sense, we pay for all our biological actions with ATP. However, in 1972, Professor Geoffrey Burstow proposed ATP and the concept of the purinergic neurotransmission. In other words, before 1972, ATP is only recognized as the energy unit inside the cell. It is one of the most important and major energy source of the body. But after 1972, when ATP is released into outside of the cell, it functions as neurotransmitter and so on. ATP can be hydrolyzed to ADP, AMP, and adenosine by CD39 and CD73. And then the four purines, ATP, ADP, AMP, and adenosine, bind to the receptors, P1 receptor, A1, A2A, A2B, and A3, and the P2X from 1, 2, 3, 4, 5, 6, 7, and P12, 1, 2, 4, 6, 8, 12, 13, and P12, 14. Now, 19 purinergic receptors have been identified. But how about the purinergic signaling in the brain? In 1977, White, they record the first direct recordings of ATP release from brain synaptosome was published in Nature. That means purinergic ATP would play important role in the brain. Till now, till 2019, the first purinergic adenosine A2A receptor antagonist is defining KW6002 used for the treatment of the Parkinson's disease was approved by FDA. It also means that we will be able to find a new target and develop new drug to treat the brain disorder. But how about the role of purinergic signaling in depression? Here, we can say the variation of serum or plasma purinergic metabolism and enzymatic activity in depression. Here, you can say the adenosine increase after antidepressant treatment. Adenosine increase in adults with depression. Adenosine decrease in adults, children, and adolescents with depression. Hypertension decrease in children and adolescents with depression. But for the oxidase, increase in depression and decrease after antidepressant treatment. Uric acid decrease in depression, but increase after antidepressant treatment. For the adolescent, the enemies decrease or increase in depression and after antidepressant treatment. But how about the purinergic receptors? Here, it concludes by this paper, the antidepressant-like effect may be induced by the activation of A1 receptor and P2Y1 receptor, and inhibition of A2A receptor, P2X4 receptor, and P2X7 receptor. So far, we have learned that the change of adenosine and the purinergic metabolism and the role of some purinergic receptor in depression. Maybe we would ask how about the ATP in depression. Actually, in 2013, the Nature Medicine paper, astrocyte-derived ATP modulates depressive-like behavior, give us some answer. They found that low ATP abundance in the brains of mice that were susceptible to chronic social defect, and it was P2X2 receptor in the medial prefrontal cortex mediated the antidepressant-like effect of ATP. And then the same team started a clinical trial to confirm whether ATP intraversely will be beneficial to depressed brain. But to date, the data hasn't been published. Hope it will come out soon. But how about P2X7 receptor, which is responsible for high concentration of released ATP and is regarded as contribution to depression? And now, the clinical trial to evaluate the antidepressant effect with P2X7 agonist, GNG 541-75446, is still ongoing. We also hope it will come out soon with positive response. Here, I will show you some data in our study. In our study, we found that the release of ATP was decreased from hippocampus, and here you can see from hippocampus and PFC after full shock stimulation. It seems it is similar with the Nature Medicine paper published in 2013. But how about the response of the P2X7 receptor? We use patch clamp to record lambda and bench ATP induced currents in hippocampal C1 pyramidal neurons. The data indicates that in C1 neurons, no changes in lambda or bench ATP induced current amplitudes after TST or foot shock. But how about the astrocyte? Here, I will show you the astrocyte response. From these slides, we can say the foot shock increased the bench ATP current after foot shock stimulation in hippocampal C1 astrocyte. That means C1 astrocyte P2X7 is upregulated by foot shock. To make sure whether P2X7 plays an important role in depressive-like behavior, we IP P2X7 here. We IP P2X7 antagonist GNG 47965567 or deletion of P2X7. We use a P2X7 knockout mice and found that inhibition or deletion of P2X7 receptor restores the decreased secretion preference after foot shock. Due to P2X7 receptor expressed and different glial cell increase microglia, astrocyte, oligodendrocyte, and so on. So we use different glial cell marker to double staining with P2X7 receptor. Here, the IBA1 P2X7 immunoreactive was increased by TST and foot shock with or without minocycline pretreatment of mice. Oligodendrocyte and NG2 glial cell respond to TST and foot shock in a similar manner with the microglia did. Neither the GFAP here nor the GFAP P2X7 receptor immunoreactive changed after TST or foot shock stimulation in company with unstressed control. To answer the potential mechanism, we applied co-PCR to measure the mRNA expression of P2X7, SLAM23, SLAM25, CD39, and CD73 HM2. Here, we can see that SLAM23 and CD39 maybe would be more attention in the future mechanism study. Here, I just make a summary. That means in the depressed brain, ATP was decreased and the P2X7 receptor were upregulated. But if we use ATP or activation of P2X7 to P2X2 receptor, maybe change the depressed brain. Or if we use the P2X7 receptor antagonist, will change the depressed brain. However, we are still unable to punch the truth of pyrrolate signaling P2X7 receptor in depression. In the future, we have a long way to go. For example, how about the glial cell types? Because glial cell type has 4 types: microglia, astrocyte, oligodendrocyte, or NG2, and so on. But what's the different role of P2X7 receptor and different glial cell in depression? And what's the contribution of P2X7 to glial neuron interaction in depression? And what's the detail or precise mechanism of P2X7 receptor in depression? All of them are need detailed answers. Finally, we are on the way to enjoy our current story. I'm most grateful to my collaborators and my students for always doing me a great favor on the project. Many thanks to Professor Peter Ellis, Patria, Alex, Yan, Haiyan, Zhen Qiang, and my students Yafei, Yin, Wenjin, Jie, Jinrong, and Yang. Also, many thanks to the Silo German Center, Natural Science Foundation of China, and 973 Program of China, who gave us financial support. Thank you, everybody. Everybody is cordially invited to come to Chengdu, the hometown of Panda. I'm very much looking forward to your come soon. Thank you, everybody.
Professor Young, thank you very much for this excellent and very clear talk. So I will go on with my talk, and after my talk, we will have questions to all of the speakers.
Okay.
So I did not make a slide for presenting myself, but I think most of the Brazilian, well, many of the Brazilians know me. I'm a professor at the Institute of Chemistry at the University of Sao Paulo. I'm very much involved in purinergic signaling, and I'm the professor, the president-elect of Brazilian Purine Club. So I will now share my screen, and a moment. So just Young, do you hear me? Do you see my screen? Hello? Okay, fine. Yes. So my talk will be on purinergic control of neurogenesis and neurodegeneration in Parkinson's disease. Before I start, I would like to thank the FASP for allowing us to present this symposium. And I would also like to thank the Brazilian Purine Club. So, my laboratory is interested in studying the conversion of stem cells into differentiated neurons. So a process called neurogenesis. And this makes also part which type of neurons we'll have, yes, and how these neurons are protected against injury and against neurodegeneration, and which are the processes involved in this. So we're looking at stem cells, the differentiation, and we're looking at receptors, and in this case, as polynergic receptors. So the idea is that how, using this simplified system, we and why using stem cells. So we have basically an investigation of complex receptor networks and their effects on early development is hardly to do, and because of this we use in vitro model which reflect simplified conditions, which may be pluri or multipotent stem cells, which upon stimulation differentiate into neurons. And what I show you here is a model that we use very much for these studies, which is the embryonic mouse stem cell, which is induced by retinoic acid, and then it differentiates first into embryoid bodies, neural progenitors, and finally on day 16 to major neurons. And this embryonic stem cell line, it's excellent for doing that, since embryonic stem cells were isolated from the inner mass of plastocysts. They can be put into culture, and then they can be readily differentiated into different types of cells, like cardiac cells, neurons, fibroblasts, and so on. And what we see here is an image showing with embryonic stem cell differentiation, where we have a staining here for young neurons, TUJ1, and we see that upon induction with embryonic stem cells that they form neuronal and also, it's not shown here, glial networks. So it seems to differentiate into a brain organoid in vitro. And this stem cell line is very good because it reflects developmental stages we see here on day 4. So the cells, they express yet embryonic markers of pluripotent cells, but they express nestin, which is characteristics for neural stem cells. So the cells have been differentiated into neural stem cells when on day 8 into young neurons and immature glial cells like astrocytes and oligodendrocytes, and then on day 16 we have a differentiation into major neurons and glial cells. So what we see here, that indeed that embryonic stem cells, when they differentiate, they show calcium oscillation, calcium transients. This is shown here as using a calcium sensitive fluorescent dye. So it means that they have calcium signaling, and purinergic receptors are very much involved in calcium signaling. So coming to this, ATP is released in the nervous system, for example, by glial cells, by astrocytes, into the extracellular space, and there they activate on neurons, astrocytes, and other cells purinergic receptors. And these purinergic receptors are P2X receptors, which are ligand-gated ion channels, or P2Y receptors, which are G-protein coupled metabotropic receptors activated by ATP, and in case of metabotropic receptors also by other nucleotides. ATP is then degraded in adenosine, where it activates adenosine P1 receptor. I won't talk about the P1 receptor. I will focus on the P2 receptors. But even so, we see here P2 receptors were formed by two structural classes, P2X and P2Y, and the P2X receptors are resembled by three subunits, which may be homomeric or heteromeric, while P2Y receptors in mammalians are represented by 8 subtypes from P2Y1 to P2Y14. And most of these receptors induce intracellular calcium signaling. So what to show here, give you an idea how we come to this study of differentiation, which may have importance also in neurodegeneration. Here we show that we use pluripotent stem cells, and we see that ATP induces proliferation, mobilized proliferation, but in the beginning, it may even just amplify the stem cell pool. And in this in these undifferentiated cells, we are mostly P2Y1, P2Y2, and also P2X7 receptor participation in this activities. Now, if we go forward to the progenitor neural stem cell or progenitor cell state, when we see that ATP again triggers differentiation, not only proliferation but also differentiation, and we see that any interference in intracellular calcium signaling that would be by inhibiting liberation of intracellular calcium or by the blocking of entry of extracellular calcium by chelating calcium, as well as by an polynergic P2 receptor inhibitor, when we see that we stop BRDU incorporation and also that we stop the ongoing differentiation. We see that ATP increases the expression of nestin. This is the neural stem cell marker, as well as of neuron specific enolase as a marker of differentiating neurons. So, I won't show you all this data that we had on this, but what we figured out in a number of studies is that undifferentiated stem cells, they, in the presence of ATP, they proliferate, they form embryonic bodies, and as a major receptor which is needed for going on with differentiating, is the P2Y2 receptor, while the P2Y1 receptor is involved in proliferation. And this decision whether a neural progenitor cell will turn into a neuron or a glial cell depends here on the P2Y2 and the P2X2 receptor, as well as on the P2X7 receptor. So we could show by RNA a interference measure and following measurements that the P2X7 receptor triggers differentiation into glial cells, while when the P2X7 receptor is downregulated, neuronal differentiation is enhanced. So we see also in another work, we could show that P2Y2 receptor agonisms triggers the differentiation of embryonic stem cells into GABAergic neurons. And we published a work on this using this model for studying mechanism of Huntington disease. So we have QS-UTP is in mouse, a selective agonist of P2Y2 receptors. So, and further on, we could see that the contribution of P2X2 and P2Y1 receptor are needed for the differentiation of pluripotent cells into glutamatergic neurons expressing, for example, NMDA receptors, as well as the P2X2 and P2Y2 receptor contribution is needed for the differentiation into cholinergic neurons. So overall, what we have seen, that we have participation of various cholinergic receptors in the differentiation process. Now, however, I would like to focus a little bit on the P2X7 receptor in my following talk. The P2X7 receptor has a physiological function, which is halting or avoiding ongoing proliferation, and, for example, maintaining the pool of neural stem cells, because if you would like to have differentiation, then you would need to downregulate or inhibit this receptor. And then also there is a physiological function of this receptor at high agonist concentration. There is in adult MPC, it forms a pore, and it induces apoptosis, and this is also seen, for example, in neurons and also in by microglial cells, which express high P2X7 receptor concentrations. But where basically we have the liberation of cytokines, and this may lead to cell death. What we see here in this next slide, where we see microglial neuron interaction, and we can see that astrocytes and microglial cells, they basically liberate ATP, and high, or also liberated by dying neurons, and high ATP concentrations will activate P2X7 receptors, or for example in microglial and also in astrocytes, and will lead to the liberation of pro-inflammatory interleukins and ROS formation, promoting cell death. So this is shown again here in the same idea, and it is also a direct connection and between cytotoxic glutamate signaling and ATP P2X7 receptor signaling, and it means that when the P2X7 receptor together with glutamatergic receptors are important for the induction of excitotoxicity, neuronal inflammation, and to processes related with neuronal death. So there are the mechanisms which are of a therapeutic receptor inhibition that we could talk about. First of all, there are upon an injury in the central nervous system, there's a cytotoxic form of a receptor expressed by astrocytes and microglial cells, and by the large ATP release by dying neurons will basically activate these receptors. And the second thing is the non-cytotoxic form, which is expressed by neural progenitor cells, maybe also differentiating neurons, and it is blocking neurogenesis and neurodegeneration. So now I come to the topic that I would like to talk about is Parkinson's disease. Parkinson's disease has basically very poor treatments, mostly based on L-DOPA. There had been some efforts in cell transplantation to restore dopaminergic neurons, and also the delivery of a growth factor into the striatum. Yes, but all these results are not so much encouraging. And now the idea, what we could have by inhibiting P2X7 receptor, maybe we could promote adult neurogenesis, and we could basically have a neuroregeneration after dopaminergic cell death. And that there is neuroregeneration or neurogenesis in the striatum has been done by a seminal work of Jonas Friesen some years ago. And based on this, we chose a brain blood barrier permeant P2X7 receptor inhibitor for inhibiting the P2X7 receptor in an animal model of Parkinson's disease. You can see here. So we use a model for hemi-Parkinson, so one hemisphere in the nigrostriatal pathway is injected with 6-hydroxydopamine to induce the death of dopaminergic neurons on the side, which happens actually through ROS formation. And then we check first on a rotational test if these animals have a lesioned nigrostriatal hemisphere. So if we challenge the animals by injection of apomorphine, so we induce motoric, but the animals who suffer from hemiparkinsonism, they cannot go straight. They are going to rotate. While the uninjected animals with the toxin, or the animals with the regenerated nigrostriatal pathway, they would go straight on. And we established this model nicely, using here basically injection of 6-hydroxydopamine by stereotaxia. And then we saw, first of all, that we have an increase in the rotation, but I see that it indicates that we have a lesion of one of the hemispheres, yes? And then we can also see this by tyrosine hydroxylase staining, where we have one hemisphere, but basically which does not stain, so that we have a destruction here of the dopaminergic neurons. And we see the same thing for the substantia nigra. So we established our model, and then we went on just to justify which are the receptors that we look on. And following 6-hydroxydopamine lesion, when we can see that here is immediately downregulating of P2X7 receptors, and when we have a recovery of this receptors, the downregulation may be to the loss of tissue, but then we get on with an increase of expression of this receptor, which may be important for neurodegeneration. And now we use the animal model, which is where we inject first 6-hydroxydopamine, and then after 1 week we check the animals by the rotational test. Then they receive intraperitoneal brilliant blue G injection, BBG, which is actually a food additive and a P2X7 receptor inhibitor. And then we wait for another 7 days, yes, and then we do the second apomorphine-induced rotational test, and we check if the rotation behavior has diminished or is the same. And finally, we perfuse the animals and go for immunohistochemistry. So what we see now is, first of all, here after 7 days, we check the rotation. Then we look, we inject the animals with BBG, and then we see that the rotation behavior in this animal is diminished, so they rotate less. That means that they may have partially recovered their nigrostriatal pathway. This you can see after 7 and after 14 days. And this effect is dose dependent. So we have a very good reduction of rotational behavior at concentrations of 50 milligrams, and a total elimination of its rotation with a concentration of 75 milligram per kilogram injected into these animals. And together with that, we can also see that in the striatum, that we have a regeneration of a tyrosine hydroxylase staining with increasing concentration of BBG. And at 75 milligram per kilogram, we have a total regeneration of this. It's important to say here, this is not a neuroprotection, it's a new regeneration, because the cells in the beginning are dead. So it means that we suggest that here is a new population by dopaminergic neurons. And we see the same thing in the substantia nigra, where we have also a regeneration for dopaminergic neurons using, again, tyrosine hydroxylase immunohistochemistry. and overall, what we see in both regions, in the stratum and the substantia nigra, what we have basically a regeneration of dopaminergic neurons as judged by tyrosine hydroxylase staining. And since we know that the P2X7 receptor is involved into neuroinflammation, we also went for IBA1 staining of microglial cells, and where we could also see that with high microglial staining upon induction of the lesion, and when with the control treatment, which does not include BBG, if we use the BBG treatment, when we have a downregulation of IBA1 immunostaining, and being consistent with a downregulation of the pro-inflammatory status. So, this P2X7 receptor inhibitors may have clinical applications in the treatment of Parkinson's disease. But now, we were interested in a second receptor subtype, which is very much involved in neuroinflammation, which is the P2Y6 receptor. And what we can see, that this receptor becomes upregulated following induction of lesion by 6-hydroxydopamine. It becomes even more upregulated when the P2X7 receptor. So it means that this receptor may be involved into the neuroinflammatory status that we have in this animal model of Parkinson's disease. And for this, to study the inhibition of these receptors, we use basically a compound in color given to us by Professor Kenneth Jacobson from the NIH, which after the same, which together or after 6-hydroxydopamine destruction of the dopaminergic neurons, we inject and we look whether the inhibition of this receptor is good for treatment of hemi-Parkinson in this animal model. And we used actually two types of protocols. The first one is the prevention protocol. It's the same protocol that we had used for the P2X7 receptor. In this case, we use the P2Y6 receptor antagonist. And also, we use the second protocol where we basically, where we had a reversion of that, as we had done, for example, for the P2X7 receptor. And what you could see here is that prevention protocol, basically, what it means that first, before we inject, or together, or before we inject 6-hydroxydopamine, then we inject with the P2Y6 receptor antagonist when we prevent this. But opposite from the P2X7 receptor, we won't have any neuroregeneration by using this P2Y6 receptor antagonist. That means the P2X7 receptor we used. So that's a little bit confusion. But talk about this, where we used the reversion protocol by with the P2X7 receptor, while the P2Y6 receptor only works in the type of prevention protocol. It means that inhibition of a P2Y6 receptor is neuroprotective, while inhibition of a P2X7 receptor is neuroregenerative. So, and this we could see here just with the P2Y6 receptor, where we have also, where we have a decrease in the number of tyrosine hydroxylase positive neurons, and if in the presence of inhibitor, this reduction is less prominent, so we have a partial protection. But we see also that we have a partial inhibition of the inflammatory process, shown here by IBA1 immunostaining. And now, just to put this together, with, we looked about that P2Y2, P2X2, and P2X7 receptor that they regulate the cellular fate of neural differentiation. If cells become neurons, or if cells become glial cells. And we are looking at P2X7 and P2Y6 receptor inhibition for neuroregenerative and neuroprotective responses in a rat model of Parkinson disease. So, and finally, I would like to thank my collaborators, and most of the work has been done here in my lab by my postdoc Agatha Oliveira Giacomelli, together with other students. And I would just here also like to thank the collaborators, Professor Kageyamas in these projects from University of Kyoto, Professor Tang from Harvard Medical School, as well as the local collaborators that I have. And I would like to thank the funding agencies, FAPES, CMPQ, and CAPES of Brazil. And I would like to acknowledge my collaboration with Tissue Gnostics, which is also, I need to disclose, I'm also a consultant for this company. Thank you very much. So, first of all, I would like to thank all the speakers. And now we have a little bit more than 15 minutes for our questions. Yes? So, what I would like to, we have questions here. I think there's a question to Professor Semyonov. Hello?
Yes, I'm here.
Yeah,
so shall I?
Maybe you may read it if you want it.
Yeah. Have you evaluated whether the lymphatic function of astrocytes is impaired in aging? Well, in this particular work, we have not evaluated this function, but we think it's really affected. First of all, we know that lymphatic system is affected with aging by different mechanisms, by different means. So basically, what is lymphatic system? It's analogous to lymphatic system for the brain, which is controlled by glial cells. And the function of lymphatic system is to remove the waste. And it's involved in perivascular space, it's involved in space between cells, extracellular space, which in brain occupies about 15 to 20% of the total volume of the structure. And when the brain ages, so there are many things changing in the brain. Stiffness of the brain, for example, due to calcification. Also, the blood vessels are not as smooth as in young age, and control by astrocyte might be also affected, because we know that surface of the blood vessels is covered by astrocytic envelope. And if we imagine that astrocytes become atrophic, their ability to control blood vessels might be also affected. And definitely, astrocytes fill the space to some extent in the brain. And what I've shown in my talk, that the number of astrocytes in aging brain, as we marked them with sulfurilamine, didn't change, but the size of astrocyte changed. And that together means that the space between astrocytes would be increased, and this space would affect the lymphatic system. So there are many aspects of astrocytic atrophy which might affect lymphatic system, even expression of, for example, aquaporins in astrocytes. So maybe also we have fewer aquaporins in dystrophic astrocytes, and this is altogether would make the significant effect on lymphatic system. And if we restore the function of astrocytes, we'll not only restore synaptic plasticity, but we also might partially at least restore the function of lymphatic system, which are affected by aging. So this is absolutely wonderful question. I really like to receive it. Thank you.
So I would like to ask the audience to put their questions into the chat. No? So I would also go on with the question to Professor Semyonov. No? So where is the glial neurotransmission? Yes. No? As I'm working with polynergic receptor, where the P2Y1 receptors and they form connexins, no? And so means no? So that they have an excitation no? Which may guide, I don't know, neuronal extensions, no? And also maybe a neuronal firing. No? That is the first question. So I understand that this is very much compromised under these conditions because you get to lose the contact between astrocytes and neurons. Yeah? And the second question is, if you have the loss of a space by the astrocytes, you basically favor the invasion of microglial cells, maybe even peripheral immune cells, which just take on the space, and they cause a constant inflammation in the system.
Sure. Okay. Thank you. Thank you very much for those two questions. And I start with the first one about cholinergic receptors in astrocytes. Indeed, it's one of the most important pathways to trigger calcium activity in astrocytes. And when calcium activity in astrocytes is elevated, so they release glial transmitters. Astrocyte also modify their morphology, etc. And with aging, if we have astrocytic dystrophy, we have fewer probably receptors on them because the surface of the membrane which carries the receptors will also shrink. This is one aspect. Another aspect is that astrocytic processes retract. And when they retract, there is more space for diffusion of any molecules, of any molecule. Would it be glutamate, would it be ATP or anything? So, for example, from the source of release to the astrocyte with young brain or adult brain, the distance is shorter. And because of the shortness of the distance and the narrower space, the concentrational profile of the ligand might be higher. So when the astrocytes shrink and they give more space for other molecules, so concentrational profiles will also drop. And I think this will also affect all types of signaling between neurons and astrocyte, extrasynaptic signaling between neurons and anything. So this is the first question, and this is absolutely would be important to look at the aging. And the second question, can you remind me? It was
was about the space, that you have basically an invasion of immune
invasion for the microglia. Well,
or even maybe lymphocytes, because probably the blood-brain barrier is not as good. Granulocytes or something, whatever.
Could be. It could open space for any cells movement in the brain, including probably cells of gliomas. So if we have astrocytoma or anything, it might actually explain why we have more gliomas and glioblastomas in older age, because it's much easier to invade the brain because of this remodeling of the brain structure. However, we know that even in sort of adult brain, in a young brain, the extracellular space is quite wide. So it's like 15 to 20% of the total structure of the total brain is extracellular space, which is quite a lot compared to other organs, I believe. And when this extracellular space increases because of shrinkage of the astrocytes, so we can imagine a lot more happening there in this situation.
So one last question. We have no question from the audience. No? Maybe we are too shy. No? So I would like to ask you about the metabolic functions in aging astrocytes. No? So maybe with down-regulated energy metabolism, no? It may be from uncoupling from mitochondria, no? Or whatever, or overloading of mitochondria, resource production, no? Could account for the shrinking of the size of astrocytes.
Yes, that's a chicken and egg question, actually. What is first, what is second? But what we know about astrocyte metabolism or about mitochondria in astrocytes is that electron transport chain is much loosely organized than in neurons. So cytochromes in astrocytic mitochondria are further away from each other than in neurons. In neurons, they literally form super complexes. So, and the electron transport is very efficient. That's why oxidative phosphorylation is also very efficient. Astrocytes, astrocytic oxidative phosphorylation less efficient, and astrocytes rely on glycolysis more than neurons. And when we have some kind of metabolic changes in the brain, in the food intake or anything, so we can imagine that electron transport chain in astrocyte might be sort of loaded with electrons. So we have more reduced cytochromes, and that's what we're basically observing. So I showed in the Raman spectroscopy, Raman microscopy image. And when we have more electrons sitting in the electron transport chain, we can imagine more reactive oxygen species generated. And reactive oxygen species is also thing which is involved in signaling on one hand, but excessive generation of reactive oxygen species might be damaging and maybe the cause of astrocytic atrophy. And so this is maybe related to what we observe. Of course, this is speculation at the moment. We have some preliminary data only, which we can link by previous knowledge. And this would require additional experiments to prove and to make connections and do experiments which will definitely link one to another, I would say. But this is very, very relevant direction what we're going to take further.
So, thank you very much for this excellent contribution.
Thank you, Henning, for
now,
great questions.
Yes,
I have a question.
Okay.
To Alexei, how many percent of the atrophy of astrocyte will cause the functional changes? That means what's the morphology and the function?
Okay, so let me rephrase the question if I understood it correctly. So the question is how much astrocyte?
How many percent of the atrophy?
Yeah.
That means
what percent of atrophy? Yes. Okay. Please.
Yes. Will cause the functional changes of astrocyte.
Oh, I think the two directly link. So the any percent of atrophy would cause functional change. In fact, the function is not all or none. So if you have a slight shrinkage of astrocyte, so then you will have a slight effect on the function. If you have dramatic change in astrocyte, you will have dramatic change in the function. And this is, I think, linked. I don't know whether it's a linear link or there is sort of non-linearity. For example, if astrocyte loses 20% of the domain of the space, and then we have dramatic change. So we don't know that because we have not looked at all the process, to see where the function is dramatically affected. So far, we think it's more or less linked. So you have small reduction and small effect on the function. Also, this is again, it's my speculation about this. Maybe there is certain threshold when the function drops really significantly. In fact, what we believe going on in the brain is that astrocytes are plastic cells, like neurons. Neurons, for example, always reestablish new connections, so they can lose synapses, they establish new synapses, rewire the brain, and this way they store new information, and this way they sort of adapt to the environmental changes. Astrocytes, as a part of active milieu, they're also plastic, and they might sort of undergo changes all the time. So the new processes might outgrow, some processes might shrink, and so forth. And we might basically reverse many of those processes. For example, with aging or with neurodegenerative diseases, we observe astrocytic atrophy or dystrophy. We also see it in epilepsy. We also see it in Alzheimer's disease. So if we would be able to reverse this process by some one or another influence, dietary, lifestyle, pharmacology, gene therapy or something, so we might be able to make the astrocytes function again very well. So this is brain still have lots of plasticity.
Yeah, thank you. Yeah.
So when I would have thank you, a question to Professor Young, yes. So you showed a clinical trial, injection of ATP for treatment of depression. So the question is, so if you inject the ATP, do you want to have that it becomes transformed into adenosine? No? And by this, would you combat inflammation in the peripheral system? Or how do you think this injected ATP could affect brain receptors? And how is this injected ATP to see with the implications of P2X7 receptor in depression?
Yes, you are right. This clinical trial was performed by Professor Gao. His team just doing this clinical trial. I don't know the detail, but in my opinion, you are right, because ATP can bind the P2X7 receptor and or P2X4 receptor. The complication will be concentrated. Yeah.
Yes,
Now I have an idea. Yeah.
I know the idea now that the P2X7 receptor inhibition will decrease depression behavior because it just puts down inflammation and neurodegeneration. Yes. So now the question is, for example, if you look at this inhibitor, so you need to inject it by stereotaxia. How would you think that you could apply this inhibitor for treatment of depression? And then I'll make a second question to you, is what about acupuncture for treatment of depression?
Okay, first I answer the second question. Right now, the clinical trial also also the animal experiment show that the acupuncture works for the depression. Yes, and they also use acupuncture on the patients. Yeah, experiment or clinical trial also show that acupuncture works. Can you remind me the first question?
No, the first question was about the inhibition of a P2X7 receptor. This inhibitor, as I know, does not go through the blood-brain barrier. What I could imagine that you couple it to nanoparticles and make a intranasal application or nasal spray, and try to bring it into the brain through the olfactory system. I don't know.
Yeah, I have an idea. Yeah, okay.
Yeah,
Yes.
no, but it's an interesting thing, because I
Yeah.
think that the work that you make is very great,
Mhm.
and it's mechanistic work,
Mhm.
no idea. Maybe also talk to Kenneth Jacobsen,
Mhm.
how to
Okay.
make an inhibitor which goes directly into the brain. You may
Mhm.
try BBG in the
Yes.
animals, but I used a brilliant blue G. It's not totally selective, but it goes through the blood-brain barrier,
Mhm.
and you could just apply it by intraperitoneal injection.
Mhm. Yeah.
Maybe we could do this in collaboration.
Yeah. Yeah. Okay. Thank you. Yeah.
So I understand that there are no more questions, and I read here from Anna Davis, who's the secretary of FASP, to thank us for this amazing symposium. And I would also like, of course, first to thank the speakers for their high quality and very interesting talks, as well as FASP for the organization of this terrific meeting. And like this, the idea is to connect scientists from all over the world. In this case, there's it's high experienced scientists from Russia and from China. And because with our BRICS states also going for the application of grants to it. Yes? So in the name of the Brazilian Pouring Club, I would like to thank everybody. Yes? And I will talk to you, Young and Alexei, shortly. Thank you.
Thank you, everybody. Yeah.
Thank you. I think this symposium can be closed.